Good morning and welcome to this SIAM invited lecture here at the joint mathematics meeting. My name is Sven Leifer. I am the current president of SIAM, and it is my tremendous pleasure to introduce our SIAM plenary speaker, who is Mariel Vázquez from the University of California at Davis. And I've just been chatting with her. She's a very nice person. She's also professor in microbiology and molecular genetics at UC Davis and a faculty director of the Center for Advancement of Multicultural Perspectives on Science, or CAMPOS, at UC Davis. And this program is interesting in that they don't just try to recruit diverse faculty, but they also support faculty to acknowledge serving a diverse set of students. So I thought that's a really cool perspective. Mariel earned her PhD in mathematical biology at Florida State University. She did a postdoctoral work at UC Berkeley and won a National Science Foundation Early Career Award in 2011. Her lab is entitled Topological Molecular Biology Lab at UC Davis, and she's interested in applying topological methods to understand the three-dimensional organization of genome in different organisms. She has served our community as a committee member on the Porter Public Lecture, which is a joint AMS-MAA-SIAM lecture. She has won numerous awards, including the Blackwell Tapia Prize. She is an inaugural fellow of the Association for Women in Mathematics, and she was elected a fellow of the American Mathematical Society in 2020. Please join me in welcoming Mariel and listening to her talk.
Thank you. I guess you will put my talk up front. Okay. I guess I need to log in again. Give me 1 second. I can't really see it too. Okay. Okay, that's it. All right, thank you so much. Well, it's really a great honor to be giving the SIAM invited address at the JMM, and it's very special to me that this is happening in San Francisco. I was scheduled to give this talk last year, and due to health problems, I had to cancel, but I'm so happy to be here, one of my very most favorite cities in the world. And I'll keep my mask on. I'm immunocompromised, but I think the audio is fantastic. And if there's anything you need me to repeat, I'll be happy to do so. Thank you, SIAM, for the invitation. Thank you for the kind introduction. And thank you all for being here. I know that there's a lot of things competing for your time today. I will start by telling you a tiny little bit about myself, because this intersection of math and biology is relatively unusual. And I always get asked, so how did you come to put the two fields together? especially the type of math that I do that is coming from what was originally labeled as pure mathematics, and then molecular biology, very structural. And my answer goes back to when I was a teenager. So I was really loving mathematics and really loving biology as a teenager, and I had to go to college and pick one of the two, because where I'm from, you cannot do a double major or take classes in multiple areas. So I decided to do math with the idea of doing biology later on. And then I had a hard time understanding how I could apply math to biology. The math that I liked in college became, I was really becoming passionate about topology, geometry, discrete mathematics, and the math biology that I saw in college was more rooted on dynamical systems, differential equations. So I couldn't see how to put the two loves together, but one day I found a way, and I found this application of topology and knot theory in particular to the study of DNA. And I did my undergraduate thesis at UNAM on applications of knot theory to the study of DNA. So one could argue that that's where it all started. It started by the Witch Sumners going to Mexico City to give a series of talks, me very shyly sitting in the back of the room as a third-year undergraduate, not really understanding what he was saying, but thinking that's the most amazing thing I've ever seen, and just being fascinated by it. So DeWitt became my PhD advisor, and I went to Florida State. I got a PhD on tangle analysis of site-specific recombination, gene and XCR systems. And there you see it's already a mouthful, like PhD thesis titles tend to be. But so I learned more biology, I learned more mathematics, and it was really interesting. When the pandemic hit, I was asked to write a book chapter on my trajectory for a wonderful book called Testimonios. And I was reflecting on these early years. So I wanted to share a couple of things that I wrote there. One of them, my first years in college, I discovered that math went beyond numbers. It also consisted of shapes, colors, and movement. Observing the world around us and navigating it teaches us about shapes and about the dynamics and randomness of different processes. And this really is kind of a guiding theme in my career. And then about math and molecular biology, it was a very unconventional path. But I didn't care, and I just followed my dream, and here I am talking to you today. So it was a good dream to follow. You'll find the book in the bookstore here in the exhibit hall. I encourage you to take in a look at it, and also the AWM has put out the even quads, card decks, and all sorts of wonderful things in the exhibit hall. All right, so today, so fast forward many years, today Javier Arzuaga, who's sitting over there, and I co-lead the Topological Molecular Biology Lab at UC Davis. This is the most recent picture of our group from last summer. Since then, some people have graduated, other people have joined. It's a really wonderful group. The two faces on the edges, they were not there on that summer day, but I will talk about their work. On the right, you see Peter Liu organizing a session today, this morning, and tomorrow morning related to the topic of my talk. And Peter is a wonderful postdoc, and he's in the job market. Talk to him. And then on the left, Jacob Lusk, who has also helped us a lot. He did the undergraduate thesis with me and has stayed on to do some research before he goes to grad school. Okay, so what is our research? We are interested in several different topics. We're interested in the structure of nucleic acids. We are interested in the mechanism of enzymes that change the topology of DNA. And this picture here on the right, and that movie, and this image at the bottom give you a glimpse of what that is. I gave a plenary lecture at the SIAM annual meeting in 2018, and the talk is available online. So if you're interested in that aspect of my work, you can always go take a look at it. It's really beautiful, and it has a lot of knot theory and low-dimensional topology. You won't see as much of that today. And then we have also been very interested for many, many years on DNA packing in bacteriophages. And more recently, the molecular evolution of coronaviruses. This was inspired, of course, by the pandemic, the lockdown. What can we do to help? We know about viruses, we know about structure, we know about modeling, we know data science. Let's do something to help with this pandemic. And that led us to study the evolution of coronaviruses. I will tell you about bacteriophages today, not about coronaviruses, but Javier gave a talk on coronaviruses a couple of days ago. Now, these are the big biological themes. From here arise a lot of interesting biological questions. They require a lot of different tools to be solved. So Jacob, in this week, I said, okay, I don't want to put the list of topics. Can we make a Venn diagram or of sorts to explain the intersections of the fields? And of course, it's impossible to claim to be an expert in all of these fields together. So in a way, we are experts in balancing and bridging between fields and deciding, given a biological question, what does it need? What does that question need to find a solution, an answer to the question? So I don't know if you see my mouse, do you? Oh, yeah. Okay, there we go. Okay, so we have mathematics, computer science, physics. In the center, I realized that statistics is all over the place in everything we do because we are studying stochastic processes most of the time. And on the bottom right, I'm very proud to say that this circle has been growing as an important component in our group, and that's largely thanks to Javier. But we have an actual wet lab at the moment at UC Davis. I mean, we always had a computer lab, a dry lab, but now we have a wet lab, and we are doing experiments in our group. And that's kind of a dream come true, because as math modelers, you always hope that you will get the data. And if you can generate the data yourself, and I'm not claiming that we are able to do a lot of that because it's extremely difficult, but if you can generate at least some of the data yourself and guide the experiments, it makes such a difference. It's such a wonderful thing. All right, and of course, this work is not done by me or by two people. It's not done in a vacuum. It requires funding, and it requires the hands and the expertise of a lot of collaborators and students and postdocs. And I want to take the opportunity to tell you, if you haven't gone to the MoMA, which is across the street, you don't need to pay the ticket at the bottom in the atrium. They have an incredible mural by Diego Rivera. That mural has been in San Francisco, I think, since the 1940s or 50s at the community college. So not many people saw it, but they moved it to the MoMA. It's open to the public. You don't need to pay. It's incredible. And I just realized that they're going to take it away again, I think, at the end of this month. So go. It's a 5-minute walk from here, or yeah. So go. I really encourage you to do that. Okay, the talk. This is the outline, so I already gave you the introduction, and I will spend quite a bit of time telling you about what is this idea of geometry and topology applied to DNA. Where do we see the geometry when we think about nucleic acids? What are nucleic acids? I'll start with that. And then I'll touch on 2 examples of our work. One, the DNA packing in bacteriophages, and another one, which is a much more recent line of research on DNA-RNA hybrids called R-loops. Okay, so geometry and topology of nucleic acids, and this is an image of a DNA knot. It's not a drawing, it's an actual picture of a DNA knot seen under the electron microscope. It was taken by Javier many years ago when he was a PhD student, and it appears in a small piece that I wrote a few years ago, and it's free for you to use however you want it. So you can download it and use it in your talks or wherever you want. Okay, so here's the first fact. The genetic code of living organisms and of viruses is carried by very long, tightly packaged nucleic acids. I will unpackage this sentence for you, starting with the term nucleic acids, because this is a math conference and we don't all understand what the term nucleic acids mean. So in biological systems, nucleic acids are DNA and RNA. Of course, in the lab, people can manufacture other molecules that look like nucleic acids, but they're not naturally occurring. And the DNA molecule here is on the left. It tends to form a right-handed double helix. So that's your first glimpse at the geometry of DNA. DNA consists of two sugar phosphate backbones lined up by nucleotides. The two strands of the DNA have complementary bases. The nucleotides are bases. The bases are, there's only four kinds, A, T, C, and G. So in a way, the code is written based on an alphabet in 4 words. If you look at the two complementary bases, A goes with T, C goes with G, and they come together. But they don't come together in a flat ribbon. When they come together, because of the structure, the chemical structure of each nucleotide, they come together as a right-handed double helix. Some DNA molecules form a left-handed helix, but most of the DNA in your body is in the shape of a right-handed double helix with a very specific helical pitch, 10.5 base pairs per turn of the helix. So already there, you see the very first naive level of packing. You have the code and a helix. And an RNA molecule looks almost the same as a DNA molecule. It could be single-stranded and double-stranded. Most of the time, it's going to be single-stranded. And RNA has the propensity of folding upon itself. Sometimes we see double-stranded RNA molecules. Most of the time when we study RNA, we study single-stranded RNA molecule, and a big question in the field is, can we predict the secondary structure of RNA? Okay, so that's nucleic acids. And then the other part in that sentence was that the genetic code is packed in very long, or is contained in very long, tightly packaged nucleic acids. This image is an old image, but I really like it because it tells the whole story in one image. You go from the code and that flat ribbon to the double helix, and then through several levels of packing until you get to the chromosome that you saw in your biology textbook in Biology 101 or in high school biology. So when we talk about DNA knots and DNA looking like a meshed tangle, people are like, okay, but why? What about the X's in my biology textbook? Well, this is what it is. So when we think of DNA as a tangled mess, we're thinking about DNA in this realm. And within the cell, sometimes DNA is very highly condensed, and then you have the X, the chromosome, like that. But at least half of the time, it is not very tightly condensed. It is packed, it is coiled into a helix and super coiled upon itself, but it's not as tightly packaged as you see at the top left. Okay, so let me go back to that sentence. The genetic code of living organisms is carried by very long, tightly packaged nucleic acids. For living organisms, the focus is on DNA molecules, and living organisms from unicellular organisms like bacteria or archaea, all the way to humans, going through fungi, plants, and all sorts of animals. The bacterial genome is one single circle of DNA, double-stranded DNA all the time. And this bacterial chromosome is tightly packaged inside the bacterial cell. If we go all the way to humans, humans have 22 pairs of chromosomes. This is a human karyotype for male, for female. The only difference is the XX versus XY chromosomes in the sex chromosomes. And this is an image of a human lymphocyte, a male human lymphocyte, showing the DNA when it's decondensed and how it forms, it organizes into chromosome territories. So let's think about this a little bit more. So again, DNA folded into a double helix. In bacteria, the chromosome is a circle, it's not a linear piece, and it is organized into discrete supercoiling domains. So the supercoiling means that the circle, the double helix, coils upon itself. And this is the next level of packing for the bacterial chromosome. So I showed you the micrograph in the previous slide here at the left. If we zoom in here and just draw it and simplify the drawing, this is what we would see, a bunch of chromosomal domains. And for organisms with linear DNA, the linear chromosomes organize into topological domains. Why am I telling you this? I'm telling you this to argue that DNA in nature is often found in circular form or as structures topologically equivalent to a circle. So if you are topologically minded, and especially if you have studied knot theory, you're like, oh, I have a circle embedded in three-dimensional space. This circle could be knotted. And I'm intrigued by those knots. So is the DNA going to be knotted or not? And the answer is yes, because knots are inevitable. Now, does a cell want DNA to be knotted? Probably no. And it finds ways of preventing DNA from being knotted. But if you just put that cable in your backpack or those headphones in your pocket, they come out knotted. And that's due to, well, it's a mathematical truth in a way. Long polymer chains tend to be knotted. With increased length, the probability that a random walk or polygon contains a non-trivial knot goes to 1. This is a conjecture known as the Frisch-Wassermann-Delbruck conjecture. These people issued this conjecture in 1961. If you look at the date, you realize this is less than 10 years after Watson and Crick published their paper on the structure of DNA as a double helix based on the images of Rosalind Franklin and Watkins. These meant a revolution in molecular biology. DNA is a long biopolymer, and they were thinking, okay, what happens to a long biopolymer? In one of the, so there's a seminal paper of Watson and Crick. There was another paper in that same issue, and in that other paper, they say, well, DNA has to be copied. And it seems inevitable to us that it will be hopelessly tangled in the process of getting copied. Somehow the cell overcomes that hurdle. We don't know how. So people were thinking about this. Long DNA molecules having to be copied all the time, but you have a helix, and if you copy a helix, you will get a tangled mess. How is that happening? So this conjecture is one of the consequences of that thinking. The conjecture was proved for polygons by Sumners and Whittington in 1988, and simultaneously by Pippenger in 1989. They were working on it at the same time, so the dates don't really mean much. It's just a time to publication. And then later for another polymer model by Diao. There's been some more recent proofs for diagrammatic models of DNA, for the Petaluma model by Evan Zohar and Haas and others in 2018. And my PhD student, Sean Witte, proved the conjecture for grid diagrams as part of his thesis in 2019, and the paper will come out this year, I hope, but it's in process. Okay, so these proofs, except for the diagrammatic proofs, these proofs rely on embedding the these polymer chains as polygons in 3 dimensions. We tend to work with polygons in the simple cubic lattice. So we think of knots and links as polygons. And once you have a polygon, now that the conjecture has been proven for all these models, we can ask if P sub n k is a number of NH polygons with fixed link type k, what is the behavior? What do we know about this value? How does Pn behave as n goes to infinity? And how does the behavior depend on the specific knot type? And I just want to put a plug here to another piece of work by my former PhD student, Sean Witte, and other people in my lab, that we took the knot table from the back of your favorite knot theory book, I mean, in particular, Rolfsen, and we said we work in biology, in chemistry, chirality is important to us. So we redrew the table so that what we see on the table are the most positive knots and links, with our definition of most positive. So if you're interested in that, you can download the tables from our website or from the paper. And I don't know if the presentation will be available, but I put a lot of hyperlinks that people can access the references if they want to. Okay, so with the conjecture proven, other conjectures arose, and now the interest moved to understanding the behavior of these knots, the asymptotic behavior. And Orlandini et al. Issued a conjecture on exponential growth rate of the number of lattice polygons with a given knot type k, and how it relates to the number of unknots. We provided recently the first proof to this conjecture. There's a paper, Beaton et al., and it's been under review for a while, but hopefully it will come out soon. It's in the archive. And I just want to give you a glimpse of that. Our proof is for NH polygons in a lattice tube. So you think of a lattice tube as the 2 by 1 rectangle times infinity, and the polygons are, all the vertices in the polygons have integer entries. So the polygon has to be embedded inside that very long tube. And to prove this conjecture, we needed, the conjecture is, the statement is quite technical, so I'm not giving you the statement, but there's an upper bound and a lower bound, and essentially this conjecture required, the proof required all these people because it requires complementary expertise coming from knot theory, low dimensional topology, that would be Shimokawa, Ishihara, and from combinatorics, polymer physics, statistics from Beaton, Atapur, Eng, Soteros, and me, kind of in the middle of everyone, helping put things together. And on the knot theory side, we come up with some new knot theory results, but basically we show that every knot or non-split link in the tube is a two-bridge knot, and that if you have a two-bridge knot like this in the tube, you can always unlink it by inserting a braid pattern. And that's a very interesting and important result. And on the other hand, for the computational physics part of it, we prove a new pattern theorem that shows that if you have a non-knot and you come up with any knot type or non-split link type that you want, you can always go from the unknot to that knot type or non-split link type by inserting patterns, one pattern for each connected component of the knot. So you can see, this is a very high above overview of the proof. It's a very long paper, but you can see how the proof would not be possible if these two communities were not talking together. There would not be a proof. There would be a bunch of examples. Okay, transition. And the other reason why I'm showing that is to also highlight that these questions in biology give rise to new mathematics. They inspire us to come up with conjectures and to prove theorems and come up with new techniques, mathematical techniques to prove the conjectures. Okay, so back to this statement. We talked about living systems and DNA. Now, let's talk a little bit about viruses. Viruses carry the genetic code in very long, tightly packaged nucleic acids, just like before. Nucleic acids, in this case, for viruses, we see DNA and RNA. I will focus today on bacteriophage, specifically bacteriophage P4, that has a double-stranded DNA genome. But there's other viruses. This is another phage that has a double-stranded RNA genome, and the human virus SARS-CoV-2 has a single-stranded RNA genome. So those are only a few examples. And if just recapping about RNA, given the RNA sequence, RNA tends to fold upon itself. So we are interested in secondary structure and how this secondary structure also is embedded in three-dimensional space to give you the tertiary structure. I will tell you a little bit about our work with RNA structure, the work of Peter, and about DNA-RNA hybrids in a little bit. Now, okay, so I hope that I've convinced you that there's a lot of geometrical thinking and topological thinking going into working with nucleic acids, with DNA molecules, RNA molecules, and biological systems. One key thing is that these are not only mathematical diversions, but the cell and the virus, they're very tightly regulated objects. And any glitch can lead to catastrophic effects. Mutations, rearrangements, alternate structures play a role in chromosomal instability and disease. Here you have an irradiated lymphocyte, human lymphocyte. The cell repairs the DNA. Sometimes it manages to repair it. Sometimes it doesn't, depending on the dose of radiation and also a little bit depending on luck. DNA replication produces replication links. If the links are not unlinked, then the cell dies. Alternate structures, so when, for example, in the case of a single-stranded DNA molecule, it may fold into something called a G quadruplex and stop a process like transcription or replication. Or the DNA and the RNA can interact with each other, which could be good or bad for the cell. So the three-dimensional organization of the genome is key to the proper functioning of viruses and of living organisms. There's geometry, there's topology, and other forms of entanglement. We're interested in all that. Coiling, braiding, linking. Other forms of entanglement arise from essential biological processes. We focus on packing, and the story about RNA that I will tell you comes from thinking about transcription of DNA into RNA. Before I transition into the examples, if you're interested in treatments for a general public of these type of topics, there's 2 number file videos in this area, the shape of DNA and how DNA unties its own knots. There's a really cool BBC podcast. Someone asks questions, and then the BBC puts together a podcast to answer that question. So here there was a person in the public asking, why do my cables keep getting tangled? And then they interviewed a few people. I was one of them. There's an article in Popular Science. When I gave my SIAM talk last time in 2018, Matthew Francis wrote an article for SIAM News. And for the Popular Science, I really like that they made these cartoons. So that's supposed to be me. And it's really cool. So I love sharing it. And there's the phages and the DNA. So you can see, it was a very lovely interview. But anyway, so there's a few things out there. If you want to read or tell someone in your family about this, these are good places to start. Okay, so DNA packing in bacteriophages. This is one of the examples. What is a bacteriophage? A bacteriophage is a virus that infects bacteria. The word phage is for eating, so it's a virus that eats. It doesn't eat bacteria, it actually blows them up. But that's where the root of the word comes from. And we are interested in icosahedral bacteriophages. These are viruses that have a capsid with icosahedral geometry, double-stranded DNA for the virus that we're interested in, is packaged inside the capsid. It has a long tail, and then tail fibers right here at the bottom that are in charge of identifying the cellular wall for the host to be infected. If the wall is the wrong wall, the virus goes away, it doesn't infect. What intrigued us when we first heard it, and this was over 20 years ago, was that DNA extracted from bacteriophage capsids, in particular these P4 capsids, is highly knotted. You're like, how? Of course, now that I told you about the Fritz-Wassermann-Delbrück conjecture, you're like, of course, it's highly knotted. But these really intrigued the biologists. How can the DNA be knotted, and how will that work out? In particular, if the DNA is knotted, the virus doesn't infect. It doesn't work. I guess viruses can get away with it because there's so many trillions of viruses in every generation that if a portion of them don't work, I guess it's fine. But, but still, this is an intriguing fact. Here is a little phage, and here's a histogram that shows if you extract DNA from these mature phages, mature means that they're complete, they're infective, you see a percentage of linear DNA, so DNA that is open, therefore not knotted or anything. You see a percentage of unknotted DNA and a higher percentage of knotted DNA. Now, I just said this, the DNA in this phage is linear, so for topologies, there's no knots in a linear piece of DNA. But this DNA has sticky ends. The sticky ends are like Velcro. So when the DNA moves around, the two ends meet each other and close. So the sticky ends come together, and now you have a circle. So now you have a circle embedded in three-dimensional space. The DNA is so tightly packaged that it's as if your computer cable in your backpack had magnets, and the two ends came together. You would pull it out, and you would see a knot. So that's what happens here. Now, if you work in the lab with bacteriophages without a tail, these are not infective at all because they don't have the tail fibers to recognize a host or the tail to inject. But they can be manufactured in the lab, and they're also naturally occurring. If you look at those tailless phages, then the knotting is off the charts. 95% of the molecules taken out of the capsid are knotted, and of that 95% of knots, more than 95% are highly knotted, meaning they have more than 10 crossings. And that's really mind-blowing and interesting to study. So the question is, what is the organization of packaged DNA inside the capsid that will yield these knots when you take the tail away? And why are the knots so complicated? Can the knots be our spies and give us information on how the DNA sits inside the capsid? And the first thing you do when you have a question like that is like, okay, well, let's open the capsid up and let's look at the knots. And the knots will tell us a story. Well, if you open the capsid up and look at the knots, this is what you see. And this is a picture that Giovanni Dietler in Switzerland took many, many years ago. We did some experiments in Barcelona with Joachim Roca. Joachim Roca sent the samples to Switzerland. Giovanni Dietler took the pictures, and we thought, okay, yay, we're going to see the pictures, but we don't. We see these rosette-like structures, and it's impossible to elucidate what knots these are. And with cryo-electron microscopy, here you'll see the credit to Tamara Cristiani a lot. Tamara Cristiani is a PhD student in our lab working with Javier and I, and she's doing cryo-electron microscopy of these bacteriophages. And you can see the DNA inside. You can see the structure of the DNA inside. But of course, this is not enough to see how much knotting or tangling is in there. So you can think, okay, mathematically, how can I model the packing of the DNA inside the capsid? If I do a completely random packing, and I take the DNA out by simulation, I see a whole lot of knots, but never as many as we see in the experiment. And the distribution of knots does not match the experiment. If I do a very organized packing, coaxial spooling packing, very, very organized, I see almost no knots. So this looks more like what we see under the cryo-electron microscope, but there's no knots. So what is the actual packing of the DNA? So let's think about how bacteriophages get packaged. When the bacteriophage is formed, DNA is packaged last. So you see at the very top, middle, these are like Lego pieces. They're different proteins that form this icosahedral capsid. And once the capsid is formed, a motor protein is adapted here in a connector region, and the DNA is pulled in. And it's believed to be pulled in some spooling fashion. Finishes the packing, some of these proteins go away, and then the tail forms. So the question here was, does the action at the motor affect the topology or geometry of the packaged genome? And this is a paper that came out in 2000. It started with the work of a master's student here at San Francisco State, where I was before going to Davis, and Brian, and he continued the work, and then an undergrad who worked with me, Sihao, and Javier, and our collaborator, Carmen Calderer. So to detect this effect of packing, we combined kinetic Monte Carlo model of the DNA packaging of the motor. This was based on a paper by Yu et al. This work was done in the Oster lab at UC Berkeley, with a worm-like chain model for the DNA. In their paper, they were interested in how the motor was working, but they were not thinking about what the DNA was doing after going through the motor. And for me, that's what was so interesting. So we said, okay, well, let's build a model. So we have a worm-like chain model with twist, electrostatic repulsions, volume exclusion, DNA capsid interactions so that the DNA doesn't go out of the capsid, and the DNA is packaged or not based on the motor model from Yu et al. Going back to, I'll go back to those simulations. Going back to the experiments, one key thing about the knots that we observe in the experiments is that the four-crossing knot is largely absent. Remember, I told you if you package the DNA randomly, you get a bunch of knots, but the distribution doesn't match what we see experimentally. When you do things at random, you would expect to see more three-crossing knots than four, and more four-crossing knots than five-crossing knots. But in the bacteriophage, you see almost no four-crossing knots. That's this band right here. So the distribution is wrong if you do random packaging. What is special about the four-crossing knot? The four-crossing knot is achiral. It's equal to its mirror image. And we have a paper that claims that DNA knots reveal a chiral organization of DNA in phage capsids. This was based on Monte Carlo simulations of polygonal chains inside a capsid, inside the box. So we up that model by doing a worm-like chain model and by following the motor model from the Yu et al. Paper. In this motor model, they worked with another bacteriophage called Phi29. DNA twists and revolves as it passes through the motor. So these gray bubbles are the motor proteins that are in the connector. So you have the capsid up here, the motor proteins, and then the tail. The DNA goes through the motor, and in their model, it twists and it also revolves in that cavity. The revolving around the inner side of the motor aperture could be right-handed or left-handed. The twisting could be right-handed or left-handed. They couldn't see what it was. They were possibly in the same direction. They have, I don't have time to show this full video, but they have a video, this is from the Bustamante lab, where they show how this motor is moving the DNA. So this is another aspect of the motor, that it pumps the DNA in bursts. So you see that it goes in, and then it kind of pauses, and then goes in again, and it pauses, and then it goes in again. Okay, so this is our version with a worm-like chain model. And the arrows are there to show you whether I'm rotating the DNA as it goes in or not. Okay, so it goes in, and it pauses, and then it will go in again in bursts, and it would pause again. There we go. Okay, and you see that the arrows are starting to disappear, so there is some rotation around the axis of the DNA. Now they're appearing on the other side. Okay, so the result here is that the average direction of coiling is biased by twisting of the DNA by the motor. In this case, instead of having arrows, I just have a line. You can see it on the left here at the stem. There's a yellow line. In this simulation, the line is not moving. And you cannot read the markers, but here I'm keeping track of time, and I'm keeping track of the amount of DNA that is being pumped in, and I'm also keeping track of two geometrical measures, twist and writhe. Okay, so here now the DNA is coiling around itself. So I changed that now I'm allowing the DNA to go around its own axis. And again, you don't see the markers, but I'll show you what they're saying at the end. So these are the final packing conformations. You can't really see a difference between them, right? They look the same. So we need math, we need geometrical measures to tell the difference. One of the geometrical measures we use is the writhe, because we know that chirality is important. And the writhe measures chirality and entanglement complexity. Here I have two trefoils. The trefoil on the left has writhe -3. Trefoil on the right has writhe, the figure-8 knot has writhe 0. And if I have the right-handed trefoil instead of the left-handed trefoil, I'll have writhe +3. Well, now look at the writhe for these two final package conformations. They're completely different. So coiling, allowing the DNA to twist around itself, yields to a very marked difference in the final conformations that can be measured by the writhe. The packaging motor induces a writhe bias in the packaged DNA conformation. Okay, and I'm going to just skip very quickly through this because I want to tell you another story before we go to questions. So our ongoing research is to go deeper on the cryo-EM that we can do in our group to extract biophysical information, such as distance between the strands, thickness of the capsid, understanding, we're studying the phages as using a liquid crystal model, so understanding which is the disordered phase and which is the ordered phase. And we also want to, Tamara, my student, wants to reconstruct the capsid and the portal proteins. Those are the portal proteins. And finally, and I'm really fascinated by this, identify the knots extracted from the capsids, getting better pictures of the knots, which we are doing, but slowly, not ready to be shown yet, and identifying those knots. Okay, so very quickly, I'm going to tell you about the next project, which was funded not that long ago, and is a fairly new project for me, unlike the phage project. And here we are working with DNA-RNA hybrids that arise during transcription. So DNA, if you know about the central dogma of molecular biology, in the central dogma, you need DNA to go into RNA via a process called transcription, and RNA into protein via a process called translation. And that's how you go from the genetic code in your cells to building the proteins that are needed to build an organism. So that's in a nutshell. So the transcription process is really, really important. During transcription, a protein called a polymerase, illustrated here by this oval, opens up the DNA double helix and starts moving and tracking the DNA double helix and making an almost identical copy of the DNA, which is this red strand. This red strand is an RNA transcript based on the coding strand of the DNA molecule. So the RNA transcript carries the genetic code information exactly as it's found on the DNA. The chemistry is a little bit different. Now, this RNA molecule is exactly complementary to one of the strands of the DNA. So sometimes it invades the double helix, forming a triple helix called an R loop. And that's what we're interested in. We're interested in asking what rules govern R loop formation. For that, we model R loops with a formal language and a regular grammar. And this is work that started through a WIM workshop, a Women in Mathematical Biology workshop organized by IPAM and by the Association for Women in Mathematics. I co-led this project with Natasha Janowska, and we were 6 women thinking about this, and we have continued. We got an NSF grant, and a NSF and IGMS grant, and it's a really beautiful, exciting project. Okay, so we build the grammar. The first stage is to build the alphabet and the dictionary. For that, we assign a terminal symbol to each half-turn of the helix, and we distinguish between stable and unstable. So remember, the double helix may be prone to opening and letting the RNA in, so we say, oh, well, that one will be unstable. Or it will be very tight and hard to open, that will be a stable conformation. For both the DNA double helix at the top here, and the DNA-RNA hybrid. And then there will be transition points. And then we issue a set of production rules that apply these production rules based on probabilities obtained by training the grammar on actual experimental data. The grant we have is with an experimental biologist who is an expert on R loops at UC Davis. His name is Fred Chedan. So in the end, we're going to have an R loop like I showed you in the previous slide, a co-transcription on R loop, written as a word in this language of alphas and taus and omegas and sigmas. And there's the R loop word. And this is what the data look like. Here, this biology group is interested in also thinking, how does the topology of the DNA before transcription affect the formation of R loops? So here, color-coded, you see that if the DNA is linear, you have the black R loops right here. If the DNA is supercoiled, the R loops are formed earlier, and if the DNA is very highly supercoiled, the R loops are formed much earlier. And let me explain that because it's not very clear. So what I'm looking at here, every line is an R loop. From, if you think of the y-axis is the DNA, I mean, is the code, the genetic code of this gene region. Every horizontal line along the vertical axis is one individual R loop molecule. And the color is telling you where the R loop begins and where it ends. So here in yellow, we see that they tend to start earlier than the black ones. So the higher the topology, the earlier they begin. So our R loop grammar is doing quite well at predicting the formation of R loops. So here again, you have the promoter region. On the x-axis, you have the gene that we're looking at. The blue line shows the experimental data. So this data tells you there's the probability of forming R loops right here, and there's another peak right there. And our result is in pink. One thing that is very interesting is that the grammar model is learning from the topology. The grammar model has no topology other than the little helix that we use to define the symbols. But we have no supercoiling embedded in the grammar, and it's performing really well as per RMSD and Pearson correlation. So that's quite interesting. And if I have, according to my timer, is 50 minutes, so if I have one more minute, do I? Okay. So we also ask a question, is there a link between secondary structure of DNA and the probability of R loop formation? And this is a question that is taking a step back and thinking, okay, let me not look at how the R loops are formed. Let me look at the transcript. Can I infer something from the structure of the transcript? And is that structure allowing me to predict the formation of R loops? And this is largely the work of Peter. That's a paper in the bioRxiv. And what we did is we defined something that we call 3 polynomial representations of RNA secondary structures. This is an RNA secondary structure. These 3 polynomial representations ignore pseudoknots, so we would remove that. And there's different ways of representing a secondary structure as a rooted tree. We took some that already existed in the literature, we defined others that suited our needs, and then we assigned a polynomial invariant. Peter had already, he had been working on phylogenetic trees in his previous postdoc. He had already developed a polynomial invariant for phylogenetic trees. So this is an extension of that work that works for these trees that represent RNA secondary structure. And these polynomials have very interesting properties. Two trees are isomorphic if and only if they have the same polynomial. So it's a complete invariant. Polynomials distinguish the RNA secondary structure represented by different types of trees. And we validated this method on a family of non-coding RNAs. And P and Q are interpretable. They're very easy to compute. So the method is efficient and accurate, so it allows for data analysis of RNA secondary structures. So when we ask this question, maybe I'll skip this part, which tells the process, but basically we predict the secondary structure of the RNA based on the DNA gene that we're working on, and we assign the graph and the polynomial to the structure, and then we compute the scale sums of these polynomial representations, and then we compare to the experimental data as we did before. These led us to observe a strong positive correlation between probabilities of R-loop formation and some of the scale sums that we proposed. So again, we are able to find a correlation, a strong correlation between the structure of the RNA transcript and the formation of R-loops, as shown in these graphs. And the conclusion that we came up, that we reached is that branches with bubbles separating short stems drive the strong correlation. And that's the end of my talk. Thank you very much. Some acknowledgements to funding, and thank you. And a reminder to go to MoMA.
Thank you, Mariel. We do have time for a few questions. So if you would like to ask a question, please make use of the microphones.
There's a question right in front of you.
Oh, right there?
Ask.
Ask your question without microphone. That's okay.
So at the start of your slide, you were talking about how there's like the DNA and like how it's twisting, and I think you said something about like right hand or something. I can't remember, but in that it was like the two lines twisting together for DNA, and in between there was like the kind of like a ladder, and they were like whole, and each one of them was kind of had different colors. Did those different colors like mean like different things for different uses of the DNA?
Yes, thank you so much for that question. So what you referred to is the DNA double helix. So each one of the straight lines is what we call a sugar phosphate backbone. And you saw little pegs that had different colors. Those colors corresponded to the bases. So the DNA carries a code, and the code is written in a very short alphabet with A, T, C, and G. So each color corresponds to one of those letters. And the letters, the way the letters are put together, tell you what the DNA is encoding for. So it tells you whether it's going to make a strand of hair or a piece of skin, so those are really important. Thank you.
Cool question. There was one over there.
Hi, that's loud. Thanks for the talk. So it seems like the things you described were all about like DNA and RNA and viruses and stuff. I'm curious, have you done things with other kingdoms of life, or do you expect the topology and geometry to vary for like plants and animals and things like that?
Well, there's inherent variations in that. For example, in bacteria, you see a circular chromosome. In the bacteriophages, they're linear, but they tend to close upon themselves. In plants, chloroplasts are circular. Even in humans, mitochondrial DNA is circular. So you definitely see variations there. And depending on how long the DNA is and how tight the confinement is going to be, you will see one conformation or another. There's some parasites, and Javier has done studies on these, called trypanosomes, where the DNA in their mitochondria consists of hundreds of mini circles, and they form a mesh. They're all interlinked, and they form a mesh. So as you point out, if you move to different organisms, each one of them probably has years of research underlying it in terms of understanding the structure. The basic structure is the same, right-handed double helix, but understanding the structure beyond that is very interesting and can vary from place to place.
Thanks.
Yeah.
Hi. Fantastic talk. A question. When you have different types of viruses that are prolate or that are very highly angular with the icosahedral symmetry, do you see different knot patterns when you do simulations for that?
Yeah, that's a very good question. Well, I mean, my mind was going to experiments when you were asking the question, and doing experiments is very hard because the genomes tend to be very, very long. Certainly, if we do the simulations on a square box or on an icosahedron or a sphere, there are some different patterns. But specifically, maybe the most important thing is the ratio between the length of the chain and the volume of the confined space.
That's really interesting. Thank you.
Mm-hmm.
Thank you. I'm dying to ask a question. Could I use your microphone for a moment?
Of course.
So you talked about DNA being sort of the storage of information for life, but computer scientists are also using DNA potentially as a storage for information because of its high density. So could you imagine using your tools to design a good storage system? What's the highest density or easily accessible or something like that? Would that be useful?
Well, I mean, I wish I could give a very informed answer to your question, because there are people thinking about this. I mean, there are people thinking about using DNA as storage devices. We have thought a little bit about using DNA and bacteriophages in particular for gene delivery. And these are things that happen, because a bacteriophage is a very effective tool that will take DNA to your organism of choice, in this case to a bacterium of choice, and deliver the DNA inside. So you can manufacture the DNA to contain a gene that you want to be inside that bacterial cell. And that's, I mean, that's been done today, and we have thought a little bit about it. But in terms of DNA computing, there are many people thinking about DNA computing. We haven't really.
Okay, cool. I haven't thought about it either. So.
Yeah.
And a question back there.
Well, it surprised me that the RNA that was tangled was actually working faster than the linear one. I mean, I would think that the more tangled it gets, the worse you know, stuff would break down. But I didn't. Do you understand what I'm saying? In your slide, when you had the super thing, he was acting faster.
Oh, for the R loops. Okay, so yeah, that's a very, very good question. Let me just show here. Yeah, you mean this one. But let me go back to the source where we explain how the R loops are formed. So R loops are formed when the RNA invades the double-stranded DNA, right? So for this invasion to happen, the double-stranded DNA molecule has to be broken. It has to be, the hydrogen bonds have to be broken for the invasion to happen. So when the DNA molecule, which is the blue helix here, is highly supercoiled, it's more fragile. And the hydrogen bonds break more easily. So that's the idea, that if you have a lot of coiling, the hydrogen bonds will open up, and that will be the opportunity for the RNA to invade. Does that make sense? It is complicated, and we don't fully understand it yet. But that's I mean, I cannot claim that what I just said is the truth, but that's what we think happens. That when the DNA gets overcoiled, this is known, that if the DNA is severely overcoiled, the hydrogen bonds break. This is known. It makes sense to us that that's when this would give an opportunity. And this also relates to the very last part of my talk that I rushed a little bit, but the very, very last slide, before the acknowledgement slide, here we said, branches with bubbles separating short terms drive the correlation. If I were able to zoom into this figure, everything in red here are pieces of this secondary structure where there's bubbles, where the hydrogen bonds have been broken. So in a way, this is giving you a sense that the RNA is more fragile in those areas, and the DNA will also be more fragile in those areas, possibly, given an opportunity for the RNA to invade.
Okay, with that, I think we're done with questions, but please come up and talk to them.
Yeah, I think there's a room, 204? Okay, so there's a room set aside for questions, an extended, I guess, 20 minutes of questions, room 204. So I will go there, and if you want to follow me, I'll be happy to just talk to you.
Nothing right now until 1 o'clock.
Here? Oh, okay, so we can stay here. Okay, then I can stay here and answer your questions.
Okay.
Okay.
So, but let's thank Dr. Vázquez again for a fascinating talk.